Okay. All right, we're going to take a look up here. And, you know, if I were going to get ready for this test here, I would at least scan the chapter and look at the pictures. You get it? I'd be looking at the pictures, say, oh yeah, I know what that is. Oh yeah, I know that. I know that. And just read the captions and say, oh yeah, I know that. And I know you have some homework questions, but the test, a lot of the test is where I give you a structure and I say, what is the name of this? And it's multiple choice. Now, if it's multiple choice, you should be able to eliminate and say, oh, it can't be heptane. The longest parent change is 8. It's got to be octane. You see that? And so that's how I'm going to do that. That's a bulk of it. Now, there is a page where I just handwrite little chemicals. Like, what if I were to write something like this, where you saw a sugar connected to a sugar, connected to a sugar, connected to a sugar? What would you call that? A sugar connected to a sugar? What did somebody say? Well, it is a polymer, but you got to be a little more picky than that, because there are a lot of polymers. What would that polymer be called? Not a monosaccharide, not a di, but a polysaccharide. So, and what are the polysaccharides you know of so far? You know of starch, is that correct? Plants make starch all the time. Plant cell walls are made of cellulose, it could be something like that. Animals make starch called glycogen, it could be something like that. And when you go to college, they'll actually show you, if you take biology, they'll show you what's the difference between this polysaccharide and this one, and I'm not going to do that. I just want to tell you that they are different. Okay, now let's get to the last polymer, and then I want to show you what I think is really neat. And I remember when I first learned it, I said, "Oh, that's not worth learning." I was so wrong. And the first time I used this book, I said, that is not very important to me. And I'll get to that after the DNA. Okay, so let's talk about DNA. What kind of elements are found in nucleic acids? You have DNA and RNA, right? And they're in the cell. And what are their purpose? I know that proteins are structures and enzymes and fuels. I know that carbohydrates are fuels and structures. Lipids are like fats, aren't they? They got a lot of carbon, hydrogen, and oxygen, but not very much oxygen. And it's almost all fuel. Some of it is used for structures. But what in the heck is DNA and RNA? What's their purpose? What's the purpose of DNA and RNA? You ever thought about that? It's not used for fuel. Yes?
It's designed to
Isn't that amazing? It's amazing that a chemical's purpose is information. Information. What? You know, if I saw a CD and the order of burned out little pits and shininess, that sequence of ones and zeros can give me a symphony or rap music. It can give me both, right? It's the sequence of that 1 and 0, isn't it? And here we have 4 letters of the alphabet, and you and an ant and a mouse and a whale, you all are made of those 4 bases, aren't they? A, T, C, and G. Why aren't you a whale? It's the sequence of it, isn't it? That's the genetic code, is the sequence. So let's take a look at it. What kind of elements are in nucleic acids? CHON, of course, and then phosphorus. So that's what you get there. Now, so what you know so far, I talked about this one other day, I'm going to do it again. What you know so far is that DNA is made, I'm going to use their picture. I'm going to use a picture in the book. How about that? Okay, here, right here. Everybody see this picture? Right? Nope. Okay. What is this a picture of? Let's see, this says, oops. Oh, man. Yeah. I'm over here, sorry. Right here. So here I have a sugar. I have a sugar here. I have a phosphate group. I have a sugar, and I have a base. And here I have a sugar and a phosphate and a base. Now, if the sugar, and what do you call this 3-part building block? This is the monomer. What do you call that 3-part building block? Somebody said it. Nucleotide. Now, the nucleotide is made of sugar, phosphate, base, okay? Now, what's the difference between DNA and RNA? What kind of sugar is found in DNA? and again, I mentioned this the other day, but I'll go to another page here. What kind of sugar is found in DNA? Deoxyribose. And this is ribose. See this? And if you take away an oxygen, you deoxy it, then that's a deoxyribose. I guess. So if I show both of these on the test, and I say, which one is deoxyribose? You said, See? That's the one that has the oxygen taken away. Isn't that right? Okay, so both of these are sugars, and then you attach the sugars to a phosphate group, and all the phosphate groups are identical. Don't have to worry about those. There's no such thing as a DNA phosphate group, or those are all the same. But what's different about the base? What are the bases that you would find that might be attached here? What are the bases that might be here? Well, for DNA, you have A, T, C, and G. And here, see that? You have A, U, C, and G. It just means, God did this. He said, I want RNA to be a little different than DNA. First of all, the sugar is going to be different, and I'm not going to give you any thymine. I'm going to give you something that looks like thymine. Uracil is actually really close to thymine. I don't know if they show that here or not. Oh, here it is. Is that uracil? No. Here it is. Yeah. See these two? That's pretty close. See the only thing different? So that's why RNA has uracil but not thymine. So now, go ahead.
Sorry. For the deoxyribose, ribose, and ribose, does it matter which one of those hydroxide bonds are, like, for an example, it's the bottom right, deoxyribose is hydrogen, the bottom right of ribose is OH, but could it have been like that top right one or like the bottom left? Does it matter which OH is?
Oh, I see. Are you talking about where the phosphate group's attached?
Yeah, no.
Where this attached? Where this attached? Where this attached?
Never mind. I don't think I would explain myself.
Actually, it seems to be this carbon here for both of them. Both of them, it seems to be that carbon that's going to attach to the phosphate group. And it looks like it's going to be this carbon right here. It's going to attach to the base. So,
is there any particular reason for that? Or
well, that's a good question. That's a good question. I guess I've never read about what would have happened if the base had been attached here. That's a great question. I've never read anything like that. Matter of fact, I've never had anybody ask me that question before. Why did it attach here and not here? That's a good question. What if you tried to make your own DNA and attach this here? That's a good question. I don't know that one. All right, now, here's what messed me up a little bit. I had never built any models in high school. I didn't know what it meant, models. When you said CH4, it just said, everything was flat to me. You get that? But you guys know it's tetrahedral, don't you? Don't you? I thought you did. Okay. So anyway, when I saw this model, have you seen this anywhere in a biology book? Have you ever seen DNA look like that? I bet you have. I bet you have. Might have had a teacher that drew it like a ladder, and they said the rails of the ladder. What are the rails of the ladder made of? Sugar, phosphate, sugar, phosphate, sugar. See that? Phosphate. And when I see this model, when I twist it into not one helix, but what? And that's why they call DNA a what? A double helix. See that? Anyway, so if we unhelicize it and untwist it, it'll look like a ladder, won't it? So the rails of the ladder are made of sugar bonded to phosphate sugar. So watch right here. Watch right here. Here's a nucleotide, and here's another nucleotide. And so we have to get one nucleotide to attach to another. Well, what is it? It's the phosphate group, sugar, phosphate group, sugar, phosphate group, sugar, sugar. Where were my bases? They're over here. They're sticking out. So when I see this model, what does the ribbon represent? What does the ribbon represent? Sugar, phosphate, sugar, phosphate. Well, where are my bases attached? Who are they attached to? They're not attached to the phosphate. They're attached to what? The sugar. The bases are attached to sugar. So where are they at? They're not the rails of the ladder. They're the rungs. Now, Watson and Crick, they won a Nobel Prize a little over 55 years ago, for discovering this structure. And they weren't great at chemistry, but they went and found some people they knew that were good in chemistry. And they even built some little models of adenine, guanine, cytosine, and thymine, and said, when I put these together, what do you think will happen? What do you think will happen here? And right away the chemist said, well, you're going to get hydrogen bonding if you have adenine and thymine. You're going to get a lot of hydrogen bonding. that would keep them together. They also knew that a piece of X-ray evidence by Rosalind Franklin, who they never said anything originally, and she was left off of the Nobel Prize originally. She did get one. She might have been dead. I'm sorry, but that was not right. They didn't have permission for her. She was a great X-ray crystallographer. She could take anyway. So some of that picture said, I think this, whatever this is, it seems to have the same diameter all the way through it. And when you put adenine with adenine, it would be big or be too small. And that meant that the DNA would have to go like this. But when you put adenine and thymine, it was the same distance as what? Guanine and cytosine. And that's how they won the Nobel Prize. A guy named Linus Pauling, who already won a Nobel Prize in chemistry, he was well known. Linus Pauling, I don't know if you heard him, but he was doing the same thing. But he said, my theory is that DNA is, the bases stick out. They have to stick out or else you can't access them. So all of his things talks about DNA having excess, but what they found out, DNA can unfold, unzip. And that's how you can have access to the bases, even though they're not sticking out. So what holds the rails of the letter together? Really strong covalent bonds. Don't let the bases get out of sequence, you get it? But what keeps the bases together? Hydrogen bonds. They aren't as strong, so if you want, you can break them, put them back together. Yes?
That was for my question. So for for replication, the helicase comes in and breaks it. So I was gonna ask, how strong is the hydrogen bond?
They're strong enough to keep it together, but enzymes can come down and go. Matter of fact, there's one called helicase, and helicase can go right down here and go zip. See that? And there's actually an enzyme that does that. Now, I'm not going to get it. If you take biology, they'll get into things where they use radioactive isotopes, and the big question was, does DNA do this? Here's DNA, double-stranded, and does DNA unzip, you make a new half, I make a new half, and the new halves come together, and you have a new DNA? Or do they come apart, you make a new half, but you stay with me? I make a new half, you stay with me. See that? And that was a big experiment they did, and they found out what happens is each half makes a new half, and there it is. So the new piece of DNA, and I have two identical pieces of DNA, half old, half new. Again, there's no new chemicals, there's no new atoms, but that was a great experiment they did. Now, one more thing they did is they found another nucleic acid, not DNA, what's it called? Now, what's the difference there? We know that DNA has deoxyribose sugar, it has these four bases. We know the base pairs are A and T, T and A, C with G, G and C, right? UNCG, A&T University. That's your memory trick. And what about ribose? What about RNA? What kind of sugar do you have? Ribose? And you just don't have thymine, you have uracil, right? Okay. And you're not double-stranded, you are single-stranded. And so A base pairs with, what's the closest thing to T? U. And if there is any kind of pairing, now they don't double strand, but here's a piece of RNA, it looks like this. It goes, whoa. So what happens is, even though it's single-stranded, this right here, base pairs here, and see that? And it makes a really weird shape. Do you remember in biology class, transfer RNA? That looks like that. A messenger RNA, that's, the COVID vaccines are made of? Messenger RNA. So messenger RNA has these bases here: sugar, phosphate, sugar, phosphate, sugar, phosphate, base. So here's messenger RNA, and make them up. Let's make them up. A, T. Oh, there's no T. U, G, C, C, A, U. Okay. So I just made a sequence of RNA. Now, where do they think they came from? The theory is that DNA will come unzipped, and now I'm not going to make DNA, I'm going to make what? RNA. And then RNA is, they call that transcription. I'm not going to ask you that question, but long time ago, when you had secretaries, I'm not trying to put them down, I'm just saying there were secretaries. And back then, the boss would call you in the office, and they want to take a message, and you had to eventually type it up, but you talk so fast. So they were actually taught how to It's called shorthand. Have you ever heard of shorthand?
Yeah.
I don't know if anybody ever does that anymore. But it said, to whom it may concern. Whoop. They have one little symbol in that, to whom it may concern. See that? So if you had shorthand, woo, man, you could write like crazy. Then you take that steno pad back next to your typewriter. Doo, 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 doo. Okay, so that's called transcribing a message. Take one code, and that's why this is called transcription, because you're taking one code and turn it into another code. Now, this code goes out of the nucleus and goes into the cytoplasm and says, ribosomes, where are you? Ribosomes, you make proteins. Come here, ribosomes. Ribosome says, I want to make proteins, but I don't know which one to make. I know which one to make. And so the messenger RNA connects with the what? Ribosome. It says, just read my message, and you'll make the right proteins. Now, DNA, Watson Crick also won Nobel Prize because of this. They thought that the message might be read one base at a time. Does you mean go get that amino acid? No, you know why? Because you could only call for 4 amino acids. And how many were there? 20. That would be a bad message. You only have 4 words in your dictionary, and I need 20? Okay, let's read them 2 at a time. AA? AU? How many messages could you have if you read them 2 at a time? Anybody know that? AA? AA? AU? AC? AG? Right? Yes?
Yes.
Now don't start with A now. What's next? How about UU? UA? See? And how many can it make? 16. I don't think the message is read 2 bases at a time, because that's only 16. And we have 20 amino acids. So what did you learn in your class?
It's 3.
3. 3 bases in a row on a messenger RNA is called a codon. And a codon says, UUA. Oh, you mean go get that amino acid? Okay. CGG. Oh, go get that other amino. Oh, yeah, I know what you mean. So they figured out that the bases are read on a messenger RNA 3 bases at a time. That was called a codon. Now, I don't know if you remember in 9th grade, there was a book, probably a page that Miss Founds gave you, and it was like a code book. Did you ever see that? It would have 64 codes in it. 64, because you know how many ways you can arrange 3 things? You have 4 things, and you want to arrange them 3 different ways, right? What is that? How many is that? I have 3 things, my fault, and I want to arrange them 4 dang it. I have 4 things, 4 bases, and I arrange them 3 at a time. What does that give you?
64.
Yeah,
Because 16 times 4 is 64.
I need to get 64.
That is 64 on 4 cubed.
Yeah. Oh, what's the matter with me? Yeah. Okay. So you get 64. So what they found out was that there are some of these codons that if you miss by a little bit, you'll still see, like AAU might be the same as AAC. And that's good. That's good. But if you have the wrong one, I'll go get the wrong amino acid, then you get the wrong protein. See that? All right. So I just want to show that to you. That's kind of the story. And here's your final story right here, is the messenger RNA leaves the nucleus and finds a ribosome. And the ribosome says, I'm going to read that message 3 bases at a time. And it says, hey, go get this amino acid. Now go get this one, go get this one, go get this one. And eventually that's how they put it in order. And then it kind of winds up on its own and it becomes a protein. Yes?
Is uracil used in RNA instead of...
Right, instead of thymine. And I didn't do that, but it's a neat little design. God said, I really don't want RNA to be exactly like DNA.
Is that just because thymine has a methyl group on it?
Yes. Yes. And if you look up there, if you look over there, the only difference between thymine and uracil is that little methyl group. You're right. So it's enough of a difference that it's not called, that's thymine, that's not thymine. Now, that's a good question. If you're an evolutionist, of course, you're going to come up with some story that says over time, thymine became uracil, and And I don't know why you'd want to do that, because it couldn't do anything for you at that time. Even if you had a million years, what would it do for you? It wouldn't do anything. If it was all designed the same way to work, that would make sense. But if you're saying evolution said, well, over time, just give enough time, that will turn into that. What was it doing in the meantime when you didn't have thymine? What was it doing? It didn't do anything. Yes?
I noticed that adenine and thymine, they're double bonds, and cytosine and guanine are triple bonds. Is that why they can only bond like C can only bond A to T?
Well, as a matter of fact, if you look at these, they have only one ring, and these are double rings. Now, I'm not your biology teacher, but some of these are called purines and pyrimidines. Did they use that word?
Yeah, yeah.
Okay, so that just means some of them are a single ring structure and some are double ring structure. And that's another thing is, if you try to put this and this together as a base pair, it would be short. If you put this and this, it would be long. And DNA has to be the same. So that's when they start pairing like thymine with adenine, and then this with guanine, cytosine. Okay. Well, anyway, that's kind of the story of DNA and RNA, and I guess that's all I need you to know at this point. If you do take a biology class and you know a little bit of organic chemistry, I think it'll help you a great deal. And for me, I just wish I would have known when they started using different models, you know. See that ribbon? I didn't know what that ribbon meant. I mean, somebody said it was the outside of DNA, but what does that mean? Do you know what it means? What's the ribbon? Sugar, phosphate, right? Okay. All right, now the last thing I want to show you is kind of a neat thing, and I'm gonna tell you that I learned a lot by teaching. I really have. It's not like when I first started teaching, I knew so much. I knew a certain amount, but I didn't know like this huge amount. And when I started teaching, I had to find out a lot of things, and people would ask questions, and I had to, look for things. Well, here's do you have this handout? Do you have the handout sheet where it had the isomers on it? On the back of this one, if you have the back of this one, I want to talk about something that is pretty neat, and I didn't. It's in your book also, and I didn't think it was that important. I had never heard of it in college. I saw it in a book, and I said, "Wonder why?" I don't know. I've never heard of it. Couldn't be that important. That was a little bit of pride there. That was my fault. But let me show you a little model here. All right, ready? Everybody here? See those? Are these isomers, or are they identical? What do you think? They're identical. Can I superimpose one on the other? They're identical, right? Is that yes? This is not a trick question. These are identical. Now, what if I replace are these identical?
Yeah.
Yes. Are you with me?
Yes.
So if you substitute one and carbon has three other things that are identical, these are identical. Let's do it again. Oh, okay, sorry. What are you doing now?
Yeah.
Identical? Sure? Can I superimpose? What do you think?
Yeah.
You think I'm going somewhere with this? What do you think? Oh, wait a minute. Okay, what do you think? Oh, wait a minute. Sorry. Sorry. What do you think? Identical?
No.
Oh. Identical? Oh. Oh. Identical? Okay, so one way of saying this is called handedness. Handedness, see that? And one of these is called a right-handed model, and one's a left-handed. Now this is a different kind of isomer because structurally they're pretty much the same, except one is a mirror image of the other. You see that? So they're called optical isomers. Optical isomers. They're mirror images. And really, I thought it made no difference. I thought, big deal. And what your book called it? Your book calls it this fancy name. What's the fancy name for mirror images? And notice it didn't happen until the carbon had 4 different things attached to it. See that? If it has 4 different things attached to it, I have to start thinking about mirror images. And it's called chirality.
Does the hydrogen count?
Say it again?
Does the hydrogen count?
Yeah. Hydrogen, my 4th. So I have 4 different things attached to carbon. See that? And so what I have here, they're not identical because I can't superimpose them. But they are mirror images, aren't they? And now you might think, well, that doesn't make any difference. And yet I was wrong. I was so wrong. And so what happened is, I want you to think of it on amino acid. See this carbon? Is that different than that?
Yeah.
Is that different than either one of those? Now, what if the R group is not hydrogen? What if the R group is this? Now, are all 4 things different? Yes. That means you're going to have a left-handed and a right-handed isomer. And again, I thought that doesn't make any difference. Let's take a look. You know, drug companies make things? They didn't know about this for a long time. And so what happened is they found out, somebody said, what? What are you talking about? Mirror images. And they have a fancy name. Let me see the fancy names on here. Enantiomers, there they are. They're called stereoisomers or enantiomers. I don't see enantiomers. Okay. Long story short. Even drug companies eventually figured out that some of their drugs had chirality. And when it takes a little more money to separate your left-handed models from your right-handed models. It takes more money. Then they found out, you know what? Only the left-handed isomer actually does what it's supposed to do. The right-handed doesn't do anything. So what did some drug companies said? Well, 50% of our drug doesn't do anything, but just leave it in there. Okay? And that's called a racemic mix, which means just leave it in there. And as far as we know, it won't hurt him. It won't hurt him to have the other guy. It just wouldn't do anything. But there were some. There were some that when you left them in there, they did have bad effects. So some drug companies still, if their research shows that one of the isomers doesn't do much, they'll leave it in there. And it costs less to make their drug that way, see? But if it makes a difference, they have to. They have to pay the money. Now, what I think is pretty neat is, how does this all work? I want you to imagine, and I got this, I started reading some more about this, but here's how it works. And that's a fancy name for stereoisomer, okay, optical isomer, stereo. I want you to imagine that this chemical is supposed to land somewhere on a cell where it binds, where the drug binds to the cell or something like that, and it's supposed to match just like this. B goes here, A goes here, and C goes here. Well, let's do these. Let's do these colors. Ready? Yellow, green, red. That's the way they're supposed to do it. And you have this in there with it. Now what? Yellow. Oh, darn. Now what happens? What happens now? It's like, now what? Now what? Now I can't do it, can I? I can't do yellow, green, red, can I? And so when it tries to bind to where it's supposed to do its thing, it can't, can it? So this is a different kind of isomer. It's called an optical isomer. And so here's what's going to ask in the, if I had a homework question on this, did it ask you about which carbon was chiral? Do you have anything on that? Okay, this is the last thing I'm going to do. I'm going to stop, okay? This is the last thing in the chapter. I'm going to stop. There you go. It says chiral carbons in carbohydrates. What? A carbohydrate can be chiral? All right, how do you know that? Let's take a look. Which of these carbons is chiral? Now this is actually kind of easy, but all I want you to know is if you ever do get into drug manufacturing or amino acids or, well, you know what they found out, amino acids? What's interesting? They're, except for glycine, they're all chiral. You see that? All of them have 4 different things attached. And you know what they found out in nature? Like over 90% of one kind of amino acid is made naturally, the left-handed one. And so if you're going to try to make your own amino acids, you have to try to, in a chemical like a test tube, you usually make half or half. And so they have to try to make just the one kind. Now, I forgot to tell you about why it's left-handed and right-handed. Okay. I'm not trying to bog you down. All right. They have a way of looking at light. You know what light is, right? Light is it going in every direction. But you can have a filter. Some of you have sunglasses called polar. You have polarized filters? Okay. Listen, I'm really a very honest person, but last time I went to a 3D movie, they're supposed to put the glasses back in there. I didn't. I had to have one. But what happens is, this is a polarizing filter, and there are 2 movies showing at once. And if you don't have the glass on, it's okay, kind of burnt. Okay. So one filter, imagine it has slits like this, and it allows light going in this direction, and that you get to see that movie. This one has slits like this, and it allows you to see the movie in this kind of light here. See that? That's kind of an analogy. So this eye is seeing this movie, and that camera saw it this way. And then the other camera saw a slightly different angle. See that? And that gives you a 3-dimension. That's why they look like 3D. Because this eye gets one, see my finger? Do you know that this finger sees a slightly different picture than, see? This eye gets to see a little bit more of that finger. Oh, this eye gets to see, anyway, that's the theory of 3D. So what happens is, these are polarizing filters. Now, if I let light go through here, light going in every direction won't go through here. Like one of them, only this kind of light go through. And only this kind of, okay. Now, what would happen if I put them together? Okay, now, can everybody see that?
Yeah.
Watch what happens. You're gonna say, ooh. Okay, I'm gonna rotate it. Oh, you're not gonna say ooh yet. We're getting there. Let me turn it down. See, if they're lined up, light will pass through it. But what if I put one at 90-degree angles to the other? Oh. Okay, so what they do is they like to use polarized light and like to shine it at these chemicals, okay? Light's going in the chemical like this, and normally it would go straight through. But these chemicals, some of them tend to bend the light to the right. And some of them like to bend the light to the left. And that's how they came up with these left and right things. And they use what they call polarized light. They do this with geology. They take rocks, I know you don't believe this, but take rocks and they have a saw that can cut it thin, really, really thin. And so a geologist can look at a rock and tell you what it is, but they can also have these samples and they put it under polarized light and they rotate it. And if they get a certain color and another color like that, oh, that's quartz, oh, that's smoky quartz, or that, anyway, they can actually identify things with using how much they had to change the angle to see something. Anyway, that's kind of neat. You can do this with sunglasses. If you have 2 pairs of glasses, or they're broken, if they're broken, and they're polarized filters, take them and just rotate them like that. It's really neat. It's really neat. So you should try that. All right, so here's to the bottom line. For some of your, I understand some of your bottom line. I got that. Let's do bottom line. If I ask a question about which carbon is chiral, let's try it. Hey, are you chiral? Well, I'm attached to 1, 2, 3 things. Well, no, I have to be attached to 4. So I'm not chiral. Are you with me? You have to be attached to 4 things. 4 different things. All right, now, what about you? I'm attached to this weird guy, and I'm attached to this one, and I'm attached to this one, and this doesn't look like any of the 4. So is that different than that? And is that different than any of them? Is that, whoa, all 4 things that are attached to you are different. What does that mean? Chiral. How about the next carbon?
Could you just get this screen to this?
Oh, I'm sorry. What about the second carbon? What's attached to you? All of this. Does that look like that? No. Does that look like this? No. And what about this right here? That's different too. This carbon has this, this. All 4 of those things are different. So guess what? I'm chiral.
So
Yes.
you count everything on either side of the element, or either side of the carbon?
Yeah, if I said what's attached to that carbon, you know what's attached to it? All that.
Okay.
What's attached? And that's all attached to that carbon. That's attached to the carbon. That's attached to the carbon. All that's attached. That's different, isn't it? How about this last guy? Are you different than that? Yes. Are you both different than all of that? Yes. And is this different than all? Yes. It's chiral. How about this last carbon? I'm attached to this whole thing. What else am I attached to?
2 hydrogen.
An OH. H and H. Oh, 2 H's are identical. See? So you're not chiral. Because you were attached to this object. An OH, that's different. H, that's different. What's the 4th thing you're attached to? Another H. You're out. All right. So then you won't have, it's not chiral. So let's try, let's do some more. Let's do one more, and I'll bet you're going to get it. Ready? Look at that picture right there. Is this carbon right here chiral? 1, 2. No, it's only attached to 3 things. You got to be attached to 4. 4 different things. You get it? Okay, now, so you're not. What about you? Who are you attached to? I'm attached to this, 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 and that. What do you think? Am I chiral? Yes. How about this one? I'm attached to this, 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 and this. Yes. What about the last carbon? I'm attached to this, 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 and what do you think? Okay. Anyway, I don't mean last carbon. I mean this carbon right here. And then the last carbon, though, is not chiral. Anyway, I just thought I'd throw it at you because I really didn't think it was that big of a deal. And yet I found out about the pharmaceutical companies, and that's a big deal. That's a big deal for them because I'll show you one you're not going to like. You're not going to like this example, but oh, shoot. Let's see it. I remember when I was in college, they talked about this chemical that when people were pregnant, women were pregnant, they sometimes had trouble sleeping. And so they had this thing called thalidomide. Thalidomide was a great sleeping tool. And then they found out, and this was bad news for that company, they found out later that the women who took thalidomide, a fairly high percent of them had babies that had arms that were shorter than normal, legs that were shorter than normal, shortened limbs. And they said, what's that all about? And they linked it back to thalidomide. So one of the optical isomers of thalidomide, it's great at a sleeping aid. What would the other one do? It caused malformations in the fetus. So have you ever heard of thalidomide babies? You have? Okay. Anyway, it so happens that there were 2 different isomers, and the company didn't know that there was a left-handed and a right-handed, and they had a mix. It was a mix in there, and that's what happened. It was sad. It's a sad story. Okay, I think you're done. I'm probably done. What do you think? What I'd like to do is you can just kind of relax a little bit, but on Monday, what I'd like to do is the entire period, if you just want to sit together, you want to look at the packet that I gave you, you want to look at whatever, and then if you want to relax a little bit, but then you're going to have to crank it up a little bit on Monday, aren't you? All right. All right, thank you very much. That's, I think you seemed like you were sort of interested in that. I don't know about, anybody want to look at the 3D glasses?
Yeah.
You want to do that? Anybody want to see these? You want to try it? So, same thing with polarized sunglasses are, they only let, you know how they get the glare down, they only let certain kinds of light through, and that's why you don't have as much glare with the polarized sunglasses.
May I get a restroom?
Yeah.
May I get as well?
What now?
May I get a restroom as well?
Okay, I'll do it two at a time. I can do two at a time.
Mr. Green, this is what I was trying to ask earlier about the H and OH. Like, does this OH have to be the same? Does it have to be that H? Or could
Hey,
it
Tim, wait. I already have two people gone, so just two at a time, okay?
Like, does it have to be that bottom one, or could it be this H? Or
Okay,
this H? 'Cause
now-
I know what you're
What?
saying with like.
Yeah, I get it. Now, is there another sugar who has an OH or has it somewhere else? And there probably is another sugar, but it just so happens that ribose is the one they found in RNA,
Okay.
and it just so happens that deoxyribose. So I agree. I think you could have an H here. It'd just be a different sugar.
Okay.
So
I
then
was
it-
just curious.
And then what you're thinking about is if I have an H here, wouldn't it have the same formula? Yes, but it'd have a different structure.
Yeah, okay.
So it'd be an isomer. So what would happen is it'd be a different kind of sugar.
Okay. Also,
Go ahead.
Sorry, go ahead.
No, I'm done.
There was. Sorry. Do you mind if I turn over
No,
page
no, I
here?
can't.
It was. It's this way, sorry. Right here. Okay, so it was showing the different bonds that they're gonna make.
Those
but
are
if
hydrogen bonding.
Right, but if oxygen's double bond right here, hydrogen is not. How can hydrogen
Okay,
bond?
so what happens is, I see what you're saying.
I just couldn't
I
remember
gotcha.
the
I
concept
gotcha.
of hydrogen bonding.
Whenever oxygen bonds with hydrogen, hydrogen gets the short end of the stick.
Yeah.
And when nitrogen forms it, it also gets a short end of the stick. So this would tend to be negative, and this would tend to be positive.
Okay.
Now here, the oxygen with the double bond here, it has 6 valence electrons.
Yeah.
And then when it shares these two, it has 8. But what you'll have is you'll have some electron pair here and here. I mean, wait, 1, 2, 3, 4, 5, 6. Yeah. Anyway,
the restroom?
yeah, ask Tim if he wants to go also. Now, so this oxygen here is sitting there with 2 sets of electrons pairs, and that's going to be a little bit more negative, and then this is going to be a little more positive. So that's where these are going to bond here. Now,
Okay.
here's the same thing going on here between nitrogen and hydrogen. So he gets the short end of the stick, and this
this
guy
one will
is
be
also going to
negative?
have one pair. It's going to have one pair of electrons that I don't think is going to be paired. 1, 2. Wait, so it has 7 valence electrons.
Yeah.
1, 2, 3.
Yeah, 7.
5, 6, 7. So like that.
Okay.
Anyway, so that's what causes that.
Gotcha.
And like you said, even Watson and Crick, they weren't as sure about that. But when they soon they gave it to a chemist, they said, "Oh yeah, there'd be 3 hydrogen bonds here and 2 hydrogen bonds here. If you're looking for something that would cause A and T to come together, that would be a hydrogen bonding."
Huh. So, so I think I get it. I just couldn't remember. I didn't realize that. I guess because in the past, all we've used hydrogen bonding with is water. So it's been hydrogen and oxygen. So
Yeah.
I was just unused to seeing
Or
nitrogen.
alcohols.
Yeah, or alcohol. I was just wasn't used to
So-
seeing nitrogen as part of that formula.
Right, I didn't use nitrogen back then because I just wanted to make sure you understood about water and alcohols. But
it makes sense.
nitrogen is also very electronegative, and when it gets with hydrogen gets the short end of the stick again. See, carbon and hydrogen is kind of equal.
Yeah.
That's why lipids are mainly nonpolar. They're mainly carbon and hydrogen. And hydrocarbons, they don't dissolve in water because carbon, hydrogen, no hydrogen bond at all. That's a great question you had. The other question about the sugar, I just think it would be a different sugar,
Okay.
but that's just not the one they found in DNA and RNA. So
Have they ever tried to replace? the sugar?
I bet they have. I believe. I mean, if you're trying to get your PhD, okay, and you think, what can I do that no one's done?
I mean, that would.
And I could be famous. Who knows? Maybe I'll be the first person to ever do that. I'm sure they have.
I mean, I don't know. Would it change anything? I don't feel like it would. I don't think.
I think it would. I think having an H instead of an OH in some place, it would
Yeah.
cause, okay,
Like a different shape. Is that what you're saying? Or like.
it itself would have a shape, but how it interacts with other things might cause a shape
I mean,
difference.
like the whole molecule, or like.
Right, so
if you
maybe the sugar here, if it had an extra OH on it, would it also tend to pucker and try to attract itself over here? Or,
Oh.
that's the theory is that's what happens anytime you add a functional group is you start getting more interactions that tend to bend things. Like that thing I showed you, that transfer RNA.
Yeah, you did.
It's really single-stranded RNA, but it just so happens that what if you had a uracil here, and you happen to have an adenine here? Well, it's going to make this part attract, and it's so weird. The shape is so weird, and this is like one of the transfer RNAs. It's really single-stranded RNA, but it's base pairing in certain locations, and certain locations don't base pair.
Oh.
And so it doesn't have a double helix. It only has one strand, but the strand's folded into transfer RNA.
So if hydrogen, like if it bonds in certain spots, then
Base pair, yeah.
because so they're bonding in certain spots because they're lined up. So wouldn't that like if it was I don't know if but if it was like CAG, right?
What is it now?
If it was like CAG or something like that, and then and it folded over, then if those two on either side, let's say G is what bonds. If
Right.
C and
Okay.
A aren't bonding, how does it?
That's a good question. How many of these have to base pair to actually fold it and make the fold stay?
Yeah.
I don't know that. I'd have to go back and look, and maybe it's two. Maybe two's enough.
Okay.
That's a great. Wow, no one's ever asked me that question. Maybe only one might not be enough. You know, when it moves around,
Yeah.
maybe it'll come unfolded, but it may take several. I didn't mention this, but at the bottom of this, you end up having 3 bases.
Yeah. Like a
That
tag.
the anticodon.
Mm-hmm.
So if you're really trying, if the codon says CCC, which says go bring me the amino acid called proline, if that's what on the messenger RNA, then why would this transfer RNA stop there? What would make him stop? He's the one who has proline on him.
Mm-hmm.
I call them trucks. The
Yeah.
transfer RNAs are like trucks, and he have this amino acid on here. I need the amino acid to be dropped off right here. Well, how do I know to stop here? Well, here's your codon. I would have G right here, and it would make me base pair here, and it would drop off this amino acid, then the next one, then the next amino acid. And it's amazing that they've discovered this at all.
Yeah, that's very cool.
Yeah.
Awesome. Well, thank you, Mr. Green.
Okay. That's what's interesting about our country. We, in the 9th grade, we tend to teach biology, but we haven't had chemistry yet. But if you go to Europe, biology is taught last because they think you need a little bit of physics and you need a little chemistry to really understand biology. That's why I think if you're taking college and you've already had chemistry, it makes more sense.
Parker.
The biggest thing on this unit is that acceleration speed over time.
Nothing.
Wait, very not. I was kidding.
Bye.
If an object is going, like, if you throw up in the air, it's just going down, at that curve, what would its acceleration be? Would it no, its acceleration
would still be
10 meters per second squared.
The speed would be 0.
Yeah.
But the acceleration would be 10 meters per second squared. You said.
So that if the time in free fall is twice as long, then you'll fall not twice as far, but 4 times as far. That's right.
Why did I just say that like the most complicated way of everything?
Actually, no, it made more sense to me.
How much trying to describe me? So one of the questions or the answer for Spanish was like, what did your friend do when they're like, oh, that's great, so I don't even have and then it goes, you internalize the anger and then get over it. And so I look at her like, so I hold a grudge.
A little upset, so.
I don't think I hold grudges.
Hey, if any of you once you get started, you can, okay? So if you want to get started there, and like I said, you have to have a pretty good pace, you don't want to be fast, you just have to make sure you don't go slow. So you don't have to start, but when you're ready, I have it ready back.
Can I take my test back there?
Yep.
Yep.
They're fun.
Yeah.
Yeah.
Yep. You have a calculator, George? Mine's almost dead.
Well,
Too much.
I don't, okay, I have a phone charger, is that the same? Is the calculator different? I've never had a calculator you had to charge. I've had battery operated and solar.
You said there's no trig on this one, right?
No, there's no trick.
Okay.
But I don't know if this fits.
No.
Okay, sorry. Now, if you wanna go borrow one, again, mine, who disappeared over a month ago, has never appeared. Miss Church says she has 3 black ones in her room that, maybe one of them's mine. But if you wanna go get one, I think but I used to have one, and I would loan them out. Now I don't have one to loan out. Yeah, if you wanna start now, you can, because,
It's a big test.
well, it is, in a way, yeah. But I wanna, when everybody does get here, I wanna give you some advice. You know, there's some questions they can answer just like that, and some are harder. Well, maybe you wanna skip those and come back to them, see that?
Which ones?
But wait, yeah, and it says you have 4 blockouts, but you can only use 2 in any column, see that?
Also, like.
So if you wanna use 2 in the first column and 2 in the next, then that's it. You've already used them up. If you only use 2 in the first, 1 in the second, 1 in the third, then that's your 4. So the blockouts, as long as you don't do 2 in a row, I'm sorry, you don't use 2 in the same column,
We only have 2 and we can just.
not 3 though. All right, you can have 2 in each column, but a maximum of how many? Maximum of 4.
Oh, 4. Okay.
Okay. So if you see a question and say, oh man, look at that question, just block it out.
Word.
Okay. All right, you go ahead and get started, and I forgot to push the staple button, so I gotta go staple a bunch of these. I just told them if they want to start early, they can. Now, the same advice I'm going to give them, if you pick up one of these, you'll see where it says that you're allowed 4 blockouts. That means there are 4 questions that I'm not going to answer those. And the only thing you have to remember, it says on there, you can't use any more than 2 in a column. So if you want to use 2 in the first column, 1 in the second, 1 in the third, you can do that. Everybody got that? So that's the only rule that I told them while you were gone. I also told them, I said, if you have a problem, it takes a long time, come back to it later or block it out. Because there are a lot of questions that are not that hard, and some are hard. You get one? Okay. You can plug it in back there, sit back there and plug it in.